Hi, my name is John Michael Sauer, and I'm the Executive Director of the Predictive Safety Testing Consortium. And today what I'd like to do is give the introduction for our urinary kidney safety biomarkers that are used to detect drug-induced kidney injury. This is all a part of a workshop on the clinical kidney safety biomarker composite measure. So, as I said, what I'd like to do is really summarize the work that has been done over the past several years around novel urinary kidney safety biomarkers. And these biomarkers have been developed to detect drug-induced kidney injury in both animals, so non-clinical species, as well as in humans. So within this talk, I'm really going to do two things. I'm going to talk a little bit about the Predictive Safety Testing Consortium and highlight our collaboration with the Foundation for the National Institute of Health Biomarkers Consortium. And then I'll really jump into the meat of the presentation, which should be pretty short, which will be around the development and then regulatory qualification of these novel biomarkers for use in drug development. So just real quickly, let's talk about the Predictive Safety Testing Consortium, or PSTC. Really what we're trying to do is derive translational strategies that help accelerate drug development. And what that means really is to come up with novel methodology that is within the drug safety space, which basically brings drugs to market faster because drug development sponsors can make better decisions quicker. The PSTC was formed 15 years ago. And really, what the idea was, as I said, was to bring pharmaceutical companies together, share innovation, validate different safety methods, all under the advisement of the health authorities, the US FDA, EMA, and the PMDA. And really what we found is that our corporate members really share the same goal, right? To go ahead and improve safety testing approaches within drug development. Really what we focused on as PSTC for the last 15 years is really around the regulatory qualification of novel translational, non-clinical and clinical safety biomarkers for use in drug development. So this slide really shows what we're trying to do in a very fundamental fashion, right? So what we're looking for are fluid-based safety biomarkers that can be used to accurately predict drug-induced tissue injury within humans, as well as in animals. But in animals, what we have is we have an advantage over when we conduct studies in humans, and that is the fact that we can look at histopathology. And so histopathology is really our gold standard, whereby whether we say we have drug-induced tissue injury or not. In humans, we don't have the luxury. We rarely have, target organ tissue that we can do histopathology on. And so what we do there is we use fluid-based safety biomarkers to go ahead and judge whether or not toxicity is occurring within humans and whether or not we have end organ toxicity occurring. The PSTC is set up within working groups, and we concentrate not on specific diseases or indications. Instead, what we do is we concentrate on target organs. And in many cases, the biomarkers used to understand whether or not a drug is causing injury to a given tissue have some flaws, some liabilities. And what we're trying to do within the PSTC is to basically come up with biomarkers that are either more sensitive or more specific, or in some cases where biomarkers don't exist, to come up with biomarkers that can be used within drug development. So really this outlines kind of our flow of what we're doing within the PSTC. So our primary goal, as I said, is to really qualify safety biomarkers for use in drug development. We, as I said, are developing these biomarkers to be more sensitive, more specific, and fill in the gaps where we don't have biomarkers. And our ultimate hope is that these novel biomarkers accelerate the safe development of therapies for patients in need. How do we do this? Well, we pull together pharmaceutical companies and other groups, including the regulatory agencies, to all work together to jointly contribute to these different projects, so that then ultimately we can receive regulatory endorsement around these biomarkers, and then they can be used within drug development studies. So we have many, many partners that we've collaborated with. I'd like to highlight the foundation for the National Institute of Health Biomarker Consortium, as they have played a very important role in what we're going to talk about today, and that's the kidney safety biomarkers. So let's jump right into it. Let's talk about the work that has gone into developing these novel urinary kidney safety biomarkers for the detection of drug-induced kidney injury, or DICKI. So you've seen this slide before, but I filled it in a little bit differently, right? And I filled it in on the bottom with our current standard, and that's serum creatinine. And so what we're hoping, and what we're going to show today, is that these novel biomarkers actually outperform serum creatinine. Now, serum creatinine is not a bad biomarker. In no way is it a bad biomarker. It's actually a very good biomarker to understand kidney disease and kidney injury due to drug treatment. However, it's not a very sensitive biomarker because it's a functional biomarker. And so what happens is it really takes up to 50 to 60% of loss of kidney function before serum creatinine actually responds, or actually responds, and there's a clinically significant change. So the issue is that serum creatinine is a functional biomarker, right? It's used to estimate glomerular filtration rate, how well the kidneys are actually filtering the blood, in other words. The kidneys, as you'd expect, have incredible functional reserve, because you need your kidneys, right? They're basic to an organism, a mammalian organism, actually functioning. And so therefore, these functional biomarkers, because there's functional reserve, respond very slowly, are insensitive. So if you think about it, we have 2 kidneys and we have about 2 million nephrons. We don't need 2 kidneys and 2 million nephrons to go ahead and regulate homeostasis within the body. But nephrons die over time, right? They become dysfunctional. And so therefore, we have more than we actually need. But what that leads to then is the fact that these functional biomarkers are insensitive. And so what we've been looking for is a panel of biomarkers that are more sensitive. And this is the panel of biomarkers, novel biomarkers, urinary biomarkers, that we've been evaluating and developing. Now, first of all, serum creatinine, of course, is in the blood, right? You're looking at serum, right? Here what we're doing is we're looking at urine. And so right there, that gives us specificity. If you would measure many of these biomarkers actually in the blood, they wouldn't tell you about kidney injury, right? They're actually very, very high in the blood because they're also in other organs. But because we're measuring them in the urine, we then have specificity for the kidney. And so we have cholesterol, we have osteopontin, we have albumin actually showing up in the urine, as well as total protein, NAG, KIM1, cystatin C, and NGAL. These have been the biomarkers that we've concentrated on, and I'll highlight through this presentation different attributes of those biomarkers and how they actually respond to kidney injury. So first of all, these biomarkers are found throughout the nephron, right? That functional unit of the kidney. And that's important because they're found within the tubules, and we know that the tubules stretch across and make up the nephron. And so therefore, we want to ensure that no matter where the injury actually occurs within the kidney or in the nephron, that these biomarkers are going to respond adequately. Now, interestingly, serum creatinine, just like urine cystatin C and urine albumin, as well as total protein, are actually functional biomarkers, right? In the case of cystatin C and albumin, and many of the other protein products, they're actually taken up in the proximal convoluted tubule, right? But if we have dysfunction of those tubular epithelial cells, then we see these biomarkers go up. Within the proximal convoluted tubule, we also have stress biomarkers or injury biomarkers. That includes cholesterol, KIM1, and NAG. Now, interestingly, just for an example, KIM1 will increase without epithelial injury, right? It increases pretty significantly. So it's actually prodromal. In other words, it happens before injury actually occurs. We also have biomarkers associated with the loop of Henle, osteoponin, as well as the distal convoluted tubules, that being osteoponin as well as NGAL. And so what you see is we basically cover the entire stretch of the nephron, at least the tubular epithelial portion of it, in order to understand different injuries. So let's now switch gears and talk about how we went about the regulatory qualification of these kidney safety biomarkers. And what I want to do, I'm going to give you a really high level here because our other speakers in the panel will actually go into more detail. So approximately 4 years ago, the 21st Century Cures Act was put into law, right? It was made an act. What happened was within this 21st Century Cures Act, Section 507, there was a very important aspect to that, and that was around the qualification of drug development tools. What it did was it actually codified the process by which the FDA would go ahead and qualify biomarkers, right? And you can see that operational process down below. What many of us also worked on was what the conceptual process was for how do we bring biomarkers into drug development. Well, first of all, there has to be a drug development need. Then you have to understand how you're going to use that biomarker and in what context, and that's really the context of use. So the patient population, for example, when it's actually being used and how it's going to be used within drug development studies. That then creates really the level of evidence that's needed in order for regulators to then go ahead and accept that biomarker. So of course, if your context of use is very broad and it asks for a lot, you're going to need a lot more evidence. But if it's very narrow and it doesn't ask for a lot, that level of evidence goes down. Well, if indeed you can demonstrate that the biomarker performs the way you hope it performs, and you can provide that evidence to the regulators, then that biomarker would then be qualified. And this endorsed drug development solution could then be applied within drug development. So we can also break down biomarker qualification in a different way, right? Conceptually, where there's really two fundamental pieces to it. One is the measurement technology and how that biomarker is actually measured. And we call that analytical validation. And there's a whole set of rules that have been developed in collaboration across many stakeholders of what's actually expected around that measurement performance. The other piece is the clinical validation, right? And that's really demonstrating that the biomarker does what you say it's going to do. So if it's a safety biomarker that is going to be able to indicate that we have kidney injury going on, well, we have to demonstrate that in our clinical validation. What's illustrated here is exactly how we validate these translational safety biomarkers, right? We utilize non-clinical species with prototypical toxicants to basically demonstrate that following tissue injury and histopathological data, right, that shows us how much tissue injury, that these biomarkers actually respond to that damage, right? Because what we then do is within this paradigm, we're using histopathology as the truth. And we then demonstrate that the biomarkers respond in a similar way to the truth. It's all about correlation with biomarker qualification. The other thing we do is we also measure the current standard biomarker. And what we want to also demonstrate in animals is that this current standard is is actually outperformed by these novel biomarkers, right? Or else we should just use the current standard, right? It's not more sensitive, not more specific around the context of use that you decide. There's no reason to bring it forward. In humans, the story changes a little bit because we can't use histopathology as the truth, right? Instead, what we're going to do is we're going to compare that novel biomarker's performance to the current standard, which we know is flawed, so that's an interesting comparison, but also to additional data, which makes up then a medical adjudication to understand that tissue injury is actually occurring within these individuals. So that medical adjudication could be around the fact that you know that an individual is exposed to, say, a nephrotoxicant. And so you're expecting to see this injury. And even though the current standard doesn't respond to a medically significant level, other information is pointing to the fact that you do have kidney dysfunction, for example. And so really where the comparison comes down to is around the current standard and then around the adjudication and the response of your biomarker. Now, when we're comparing the current standard, we have to be careful, right? Because anytime that we're more sensitive than the current standard, according to the current standard, we'd be a false positive. And anytime that we're more specific than the current standard, that would be a false negative. And so you have to be careful how you architect your design such that it's very well understood that there is a flaw within that current standard and how your biomarker is going to outperform it. So let me put this all in perspective of our project around the kidney safety biomarkers. And so we had a non-clinical learning phase that actually resulted in the qualification of 7 biomarkers by FDA, EMA, and PMDA. We also then expanded that qualification by using the letter of support mechanism to demonstrate that osteoponin and NGAL actually performed well in the rat, as well as in some cases in the dog and non-human primate. And so that really established that animal link, that non-clinical link to histopathology, to performance of the biomarker. Then what we did was a clinical learning phase. And this was around using a prospective healthy volunteer study and then samples from a cisplatin study where individuals with mesothelioma were treated with relatively high doses, that was the current practice back then, of cisplatin. And we basically demonstrated the baseline of these biomarkers across a large cohort of healthy volunteers, and then demonstrated that indeed cisplatin would increase the level of these biomarkers in the urine. And I'll show you some of that data in a moment. Then we had the clinical confirmatory phase, where we had 2 prospective studies, an aminoglycoside study and a cisplatin study, in which we went ahead and looked at the response of these biomarkers, comparing these novel biomarkers to medical adjudication of kidney injury. Ultimately, all this data will be submitted to FDA, EMA, and PMDA for qualification of these biomarkers. Now, one thing I must say is that in the learning phase, this clinical learning phase, we're able to get a qualification for these biomarkers to be used as a composite measure. And that's really what most of this webinar is going to be about, and discussion on how to actually use these biomarkers. Again, these were the qualifications for the non-clinical, and this was an important step. This is actually the first time that the 3 agencies qualified biomarkers for use in drug development. This was the letters of support from both FDA and EMA that introduced NGAL and osteopontin into the panel. And then this was the qualification for the composite measure for the use of these 6 novel biomarkers within phase 1 clinical trials for evaluating the cohort of subjects that are moving through that trial. Let me show you a little bit of data around how these biomarkers perform. there is some intrinsic variability, and that's what we see here from the healthy volunteer study that you're looking at. There is some intrinsic variability that are associated with these biomarkers across the population. There are some subtle differences with some biomarkers between males and females, and there are, and you can pick it apart, some subtle differences between age groups, but mostly age, didn't have a dramatic effect on these biomarkers. One thing to point out that you should notice under the biomarkers next to their name is that we're actually measuring the biomarker and then quantitating how much is actually in the urine, then dividing by how much urinary creatinine is actually in the urine. And the reason why we did this is it tightens up the variability quite dramatically, because you can have individuals with different urine volume based on hydration state. And this really takes care of that in many ways. And so what our objective is to be able, and the reason why understanding variability is so important is because what we want to do is we want to utilize these biomarkers where we measure them before an individual is treated with a potential nephrotoxicant, and then afterwards, and watching how the biomarkers respond to look at a fold change in the response of these biomarkers. Because depending on the assay that's utilized, you're going to get your absolute numbers or values are going to be different, simply because there are no reference standards for these assays yet. So that's a part of the price of novel biomarkers, right? And so you have to make do with what you can. So let me show you some exciting data here, and we'll go into this further in the webinar. Other speakers will go into this, and we'll talk about the panel session. But what we're looking at are individuals, first of all, from the healthy volunteers, and you can see during the healthy volunteers how many times these biomarkers actually deviated from what we're calling normal, and it's relatively small, right? However, what you can also then see is the number of patients that had medically significant changes in serum creatinine and how the biomarkers, the novel biomarkers, actually responded. And they respond quite well, 100% of the time, basically. Sure, there's some 95s there. However, what's really interesting in red is for individuals, patients, that do not have medically relevant changes in serum creatinine. These are the percentages where these novel biomarkers actually change, right? And we know, because of the way the study was designed, high dose cisplatin, these patients are experiencing some type of nephrotoxicity. It's just not enough to be detected in a medically relevant way by serum creatinine. However, it can be detected with these novel biomarkers. And so that's the real exciting part, right? Sensitivity. But, if we're just comparing the serum creatinine, these are all false positives, even though, from the animal data, right, the non-clinical data, we know that these biomarkers outperform serum creatinine. So what we're working towards now is a set of prospective studies that I've already described. And at some point, we'll go ahead and also receive a qualification for these prospective studies. Right now, we're in the middle of basically going through, the final process around the validation of the assays, and then the formal adjudication process, and then move forward with the agency around bringing this qualification forward. So what happens after qualification? And I think this is an important thing to think about, right? We know these are novel biomarkers, very much like a phase 3 study. It's a relatively small data set that you're making decisions around the performance of the drug in phase 3, the biomarker here in qualification. So we know we're going to learn a lot more after these biomarkers are used more broadly. And so what we've done at CPATH is put together the biomarker data repository, or the BMDR. And what the objective is to really capture our experience with these biomarkers, both in the clinical as well as non-clinical space, and then allow stakeholders to be able to use this data to make better decisions around the biomarkers. And what I hope happens is that really the way we're going to use these biomarkers is going to change over time as we learn more. right? We'll be able to better optimize how we're applying these biomarkers in drug development studies. So a couple quick concluding points. I think we've made significant progress on both the development and the regulatory qualification of our novel urinary safety biomarkers for the detection of DICKI in both non-clinical species and in humans. We are facilitating the use of these biomarkers, and it's really a primary goal for PSTC. It's really the reason why we put on the webinar today, right? To be able to, educate folks on how to best utilize these biomarkers, and the fact that we've collected all this data and that these biomarkers more or less are ready to be utilized. Another goal for the PSTC, and I didn't get into this very much, is to go ahead and compare and contrast the different analytical methodologies that are available to measure these novel biomarkers. We're doing that in conjunction with the US Pharmacopoeia in order to basically level set and understand where the differences are in these assays. And then finally, what we want to do as CPATH, as PSTC, is to be able to facilitate data sharing and share the experience of how to use these biomarkers, through both BMDR as well as other means. So with that, I'd like to thank everybody and hopefully see you at the webinar.